Hello, this is our pre-lab discussion for Module 4 in CHY 114, our dissolved oxygen unit. And so remembering our Green River project, that we are investigating the water quality issues along the Green River. And so far in modules 1 through 3, we've really been focusing in on looking at specific pollutants and looking at specific uh, issues that might be affecting the water quality. Today we shift that focus a little bit and we start to look at what those pollutants might be actually doing to the river, what some of the issues are that they might be causing. Because if you remember at site C, down here at our final stop downriver, so to speak, it was observed that there were a lot of fish kills, or a lot of there was a lot of fish kill in general here at this site. And so that's one of the things that we're investigating, is to try to see what might be causing that. And one of the potential issues could be the level of dissolved oxygen. We've talked about eutrophication a little bit, and hopefully you've looked up eutrophication, done a little bit of background reading on it. But eutrophication in general is when a water body receives an influx of nutrients. Things like nitrogen and phosphorus are entering into a water body in excess compared to what normally comes in. And those nutrients can then cause a pretty big spike. It can cause an increase in the amount of phytoplankton. Phytoplankton then giving rise to algae blooms. And what can happen then, or what does happen then with the algae bloom is, sure, initially we can get a sort of, sometimes you see sort of a spike in dissolved oxygen levels because there's more photosynthetic activity with extra algae in there. There's more photosynthesis happening when sunlight hits the algae. As time goes on, though, the algae dies, so typically settles out, and then the decay process uses up oxygen, uses up a lot of oxygen. And so we can end up, when that happens, with severe drops in our dissolved oxygen levels in a water body, which can be disastrous for fish populations. We can see here some typical levels of dissolved oxygen. If we're up around 10 ppm, 10 parts per million, 9 or 10, we're fine. That's plenty of dissolved oxygen for fish to survive and thrive and live perfectly happy, peaceful little fish lives. When we start getting down into the range of 6, 7, 8, somewhere in here, now we do have enough oxygen for spawning and growth, but we're starting to get down to a point where populations are not going to thrive. They're not going to really be able to exist in large numbers at those oxygen levels. We start getting down to 4 or 5 ppm. Now we start actually seeing stressful situations where populations might decline because there's simply not enough oxygen for the fish to survive in a healthy manner, to be able to then, reproduce and so on. And then even below that, we start getting down to dissolved oxygen levels below 4. And at that point, that's just too low. Fish are not going to be able to survive. And you're going to start to see lots of fish kill like we saw at Site C, perhaps. And so that's really what this lab is all about, is trying to see what is going on with the dissolved oxygen in the Green River so that we might be able to determine if the oxygen levels are contributing to those fish kills or not. And so to study this, to look at our dissolved oxygen levels, we're going to use something called the Winkler method. This Winkler method is an analytical technique. We're going to use titration, which is something that we haven't used before. The Winkler method is an incredibly accurate method for finding dissolved oxygen. Sure, there are, quote, easier ways we can get dissolved oxygen probes, where we just stick a probe right down into the sample, and it'll read our dissolved oxygen, spit that out onto a computer for us. Simple and easy. This method that we're going to use today, though, is more accurate. This method is actually used to calibrate those probes to make sure that they're working properly. These probes can measure to about 1/10 of a ppm, whereas the Winkler titration can measure down to 5 hundredths of a ppm. So it's an incredibly, incredibly accurate analytical tool. And so let's look at what's involved. These are the equations right from your lab manual. But what's first done, typically right out in the field, when the dissolved oxygen, or when the samples are taken, you take a sample of water, and then almost immediately add in some manganese hydroxide. And so again, this is typically done right in the field. Oops. What this does is it's what we call fixes the oxygen. And so any of the oxygen that is present is going to react with this manganese hydroxide to then form this manganese oxyhydroxide complex. And so then at that point, the sample would be sealed so that no more atmospheric oxygen can get in. And again, any of the oxygen that was present is now going to be locked up in this complex here. And then we send it through another series of reactions. We add some acid to the sample, forms this manganese sulfate. And then we add some I-. And what we end up doing there, what we're really concerned with, so to speak, through our course of reactions, is then the formation of this I2 here in equation 3. This I2 is very important for us because that I2 is what will actually react for us in the titration that we complete, because we then will react sodium thiosulfate. And really it's the thiosulfate ion itself, the S2O3 2-, then reacts with the I2 to form this S4O6 and I-, but honestly, we're not concerned with the products of this reaction. We're not going to do anything with those. What we're concerned with is the stoichiometric amounts involved in this reaction right here. We're concerned with being able to add enough thiosulfate to react with all of the I2 that's present, because we have a very convenient indicator that tells us when this reaction is complete. We use a starch indicator, which will turn blue when all of the I2 is gone. And so in other words, when we have added exactly enough S2O3 to react with all of the I2, the reaction turns blue. It tells us the reaction is complete. It tells us we can stop our titration. We can stop adding S2O3 into our reaction. What we can do then, so again, sort of keeping in mind what this indicator, what this endpoint tells us, is that at this point, when the reaction turns blue, we have added exactly enough S2O3 to react with all the I2 that was present. And so if we know how much S2O3 that we had, if we know how many moles of S2O3 that we added at that point, we can compare that then. We could just use our stoichiometric ratio, our 2-to-1, to compare that to how much I2 had to have been present in our sample. We then relate that back using equation 3 to how much manganese sulfate had to have been present. So again, we're just saying, okay, I know how many moles of S2O3 I had to have added. That will tell me how many moles of I2 that I had. And then equation 2 tells us how many moles of manganese sulfate had to have been present. Then we backtrack again. We can relate the moles of manganese sulfate back to the moles of this manganese oxyhydroxide complex. Backtrack one more time, and we can use the number of moles of that manganese oxyhydroxide to relate back to the number of moles of O2 that had to have initially been present. This is no different than any stoichiometry problems that we've completed in lecture. The only difference is we're carrying it through more than one reaction. That's all that it is. It's still the same exact type of reaction that we've done numerous times in lecture. And so let's look at how the units of this all work out. And so again, assuming we are able to know and to calculate how many moles of S2O3 that we actually had, we can use equation 4 to then relate that back to the moles of I2 that had to have been present. And so now we know how many moles of I2 had to have been present. We can use equation 3 at that point to tell us how many moles of magnesium sulfate had to have been there. Then using our ratio in equation 2 gets us to how many moles of the manganese oxyhydroxide. And then finally to equation 1, how many moles of O2 had to have been present in the very beginning. And so using this method, again, as long as we know how many moles of S2O3 that we used, and we use that, we know that just from the fact that we use a very reliable indicator. The indicator tells us when we've added enough S2O3 to react with all of the I2. So as long as we can calculate exactly how many moles of S2O3 we used at that point, we can backtrack through all of the reactions to find our moles of O2 that had to initially be present. And then we'll talk about how we use that moles of O2 to then convert that over to the parts per million of dissolved oxygen. For now, let's just keep focus here on these equations and how we use those to backtrack our way through to find the amount of O2 that had to have been there. So you can see, though, that in order to do this, in order to backtrack our way through, we have to be able to find a way to precisely know, accurately and precisely know, exactly how much S2O3 that we used. This is where we run into a bit of a problem. It turns out that thiosulfate is what we call hygroscopic. It means water molecules love it. You get a sample of thiosulfate out, and water molecules that are around, even in the atmosphere, are just going to attach themselves onto the thiosulfate. And so that presents us with an issue that we have to overcome. And so, for example, in this lab, we want to prepare 500 milliliters. This is what task 1 is sort of all about. We're going to prepare 500 milliliters of 0.004 molar thiosulfate. And we've looked at this calculation before. And so in order to find out how much of the solid thiosulfate that we need, we're just going to take our volume that we're looking for, the volume we're trying to make, 0.5 liters, 500 milliliters equals 0.5 liters. We multiply that by our molarity of thiosulfate. And then we multiply that by the molar mass of thiosulfate. Tells us that in order to make this solution, I need 0.316 grams of my sodium thiosulfate. Now, if thiosulfate were not hygroscopic, if it didn't suck water in, then I could say, all right, great, I'm going to use 0.316 grams exactly, and that's going to give me 500 milliliters of exactly 0.004 molar. But thiosulfate is hygroscopic. It is going to suck in the water molecules. And so that tells us that when we go through this preparation, when we prepare our 500 milliliters of our 0.004 molar sodium thiosulfate, in order to make that solution, we're going to add 0.316 grams of our thiosulfate. And then we're going to add water till we get to a total volume of 500 milliliters. We've done that before. We've made solutions like this before in lab. The problem that we run into now, though, since sodium thiosulfate loves water so much, when we take the mass, when we get that 0.316 grams, we've got no clue how much of that might actually be water. Some of that 0.316 grams is absolutely water molecules that are just attaching themselves to our solid. And so we don't know how much of that 0.316 is water and how much of it is thiosulfate, which means that ultimately we don't know the actual molarity of the thiosulfate solution, because we don't know how much of that 0.316 is thiosulfate. And so we don't know the actual molarity of our solution, but the accuracy of the Winkler method relies on knowing that concentration precisely. We have to know the exact concentration of the thiosulfate for this method to work. And so this is where task 1 comes in. Task 1 is all about the standardization of the thiosulfate. It's all about once we make our solution. So at this point, we've made our 500 milliliters of 0.004 molar. We've taken our 0.316 grams, added it into our volumetric flask, add water up to 500 milliliters. And so now we have our solution. Well, we don't know exactly what the concentration is. We know that it's around 0.004 molar. Well, we don't know it precisely. And so this standardization process is going to get us that information. It's going to tell us exactly what the concentration of our thiosulfate is. And to do this, we make use of what we call a primary standard. Primary standards are ultra pure. Not only are they ultra pure, they're not hygroscopic. They don't suck in the water like something like thiosulfate does. And then to go even further, we usually dry them in an oven and then store them in a desiccator so that no water, even though water doesn't stick to it, we don't even give water a chance to get near it. And so primary standards are very often used for just this purpose, to then standardize another solution. We're using the primary standard potassium iodate. And so what we can do with potassium iodate is make a solution using the mass, take their solid, put some in to make whatever the concentration we're looking for. And the advantage is that when we do that, we can know precisely exactly what the concentration is. That's the whole idea of a primary standard, is that we can make a solution up using just the mass and using the solid amount. And that solution will be our target concentration. In this case, we're going to be able to use a solution of KiO3 with a concentration that's known down to 1/10 of a millimolar. So a very, very accurately known concentration. And so this then introduces another equation into the ones that we looked at. So equation 4, remember this equation here, equation 4 is sort of our equation of the actual reaction that we're running with our titration. And so at this point, things have already been added into our solution. And we're just now taking our thiosulfate, which is in our burette, and the actual procedural notes your instructor will talk about. We'll talk about in a lot of detail. But you're going to have some of your thiosulfate solution in your burette and be adding that. And again, when it turns blue, we know that we've added exactly enough of this to react with all of the I2. The difference in our standardization is how we're going to get that I2. So in this, in what equation 5 is all about, is using our KiO3, is using our potassium iodate, again, with its precisely known concentration, and then using some I- or some iodide ion, usually introduced with potassium iodide, just some Ki, and then some concentrated acid. And that's how we're going to then form our I2. And so then that I2 will be used in this reaction here. In this reaction, in reaction 5, the I- and the H+, our acid, those are in excess. And so our KiO3 is absolutely our limiting reagent here. And so we can use that amount of the KiO3 to tell us exactly how much I2 is going to be produced. So let's take a look at those calculations. And so in this experiment, in our experiment, we're starting with 5 milliliters of KiO3, so 0.005 liters of our KiO3. Our lab technician does a great job in making up a very precisely known concentration of the KiO3. So we're going to use 0.0017 molar KiO3. And so just that, if we kind of stopped right there, just this information here would tell us in our standardization process how many moles of KiO3 we are actually starting with. And so if we know how many moles of KiO3 we start with, then we can just use our molar ratio to figure out how many moles of I2 had to have been formed. And then if we know how many moles of I2 had to have been formed, this is what would be sitting down in your beaker when you're performing the standardization process. So now you would know how many moles of I2 that there are. And again, the whole point of our starch indicator is that it shows us when the reaction is done, meaning we've added exactly enough of the S2O3 to react with all of the I2. And so we carry this through. This tells us that when you are performing your titration, when you have your S2O3 dripping down through your burette into your flask, performing your titration, you had to have used at the end point of this reaction, when the reaction is over, based on the amount of KiO3 that we started with, you know that you have to use 5.1 times 10 to the negative 5th moles of the thiosulfate. And so this is a number that will be the same for everyone in doing your standardization process. At the end point of your standardization, when the liquid in your flask turns blue, you are absolutely using 5.1 times 10 to the negative fifth moles of your thiosulfate. And so if we know that information, and again, some of this will make more sense as you're seeing your setup, but if we know that information, we also know what the titrated volume is, how much of the S2O3 you had to have used. And so that is the number, and that is the only number in task 1 that will be different for everyone. This is your titrated volume, the average of your 3 titrated volumes. And so you're going to perform the titration a minimum of 3 times, usually 4. You're doing 4 titrations typically gets us with 3 that are very precise and accurate with each other. And so you've got your titrated volume now. You know how much S2O3 you had to have used to get to the end point of your reaction. And so we take the number of moles of S2O3 that we calculated had to have been present, divide that by our titrated volume, and now that gets us to the molarity of our S2O3. And so that tells us precisely, using a primary standard, this tells us what the molarity of our S2O3 2- has to be. This is what task 1 is all about. It is making our thiosulfate solution and determining the exact precise molarity of it, so that we can then use that solution for task 2. Because now that we know the concentration, now that we've found the standardized concentration of our thiosulfate, we can use that to now titrate our Green River samples and find our dissolved oxygen content. So again, let's just bring up equation 4. And so we know that at the end point of this reaction, when the starch indicator turns our liquid blue, turns our sample blue, we know at that point we've added exactly enough S2O3 to react with all of the I2 that was present. And so you're going to run this titration. Your procedure for the titration part of the lab is going to be pretty much identical for task 2 as it was in task 1. You're going to have your Green River samples in your flask. You're going to have your burette down in your flask, and you're going to titrate. Your S2O3 will be in your burette, and you're going to titrate that into your sample until it turns blue. And so at that point, you will have your titration volume, the volume of S2O3 that you have to use to make your reaction turn blue. And so we're going to just look at a little example of how this all works out. And so in your determination of the dissolved oxygen, so again, experimentally, you're just running a titration. And again, you're hopefully watching this pre-lab video before doing the lab. If you don't understand what a titration is, that's fine. You will. That's one of the things that your instructor will be talking about, is what a titration is and exactly how you do it. This is just sort of here we're just giving the background information and getting you ready. And so you're going to perform your titration. And then from that, you're going to have a volume. You're going to know the volume of S2O3 that you used to perform your titration. You then will multiply by the molarity. This is the standardized molarity that we found in task 1. Should be right around 0.004. Won't be exactly 0.004, but it should be in that ballpark. And so then you multiply that by your standard molarity, by your standardized molarity. We can now use all those stoichiometric ratios that we talked about in the beginning to backtrack this back to the amount of O2 that had to have been present. And so take a look at those equations again, get your lab manual out if you need to, and look at these equations. But we can see that, again, just using these equations and going from our liters of S2O3, then our molarity of our S2O3 gets us to our moles of S2O3 that we have to use at the end point of the reaction. And then we can backtrack through all the equations to eventually find the moles of O2 that had to have initially been present. And this is our target. This is the amount of O2. We still need to do more with this, and we're going to talk about how we get from here to our parts per million of O2. But this is really the sort of the guts, so to speak, of the Winkler method and of our whole process here, is that this gets us to the sheer molar amount. This gets us to the amount of O2 that had to have been present. And so we now know how many moles of O2 were in the 30 milliliters of Green River sample that we used. So remember, your procedure calls for you to use 30 milliliters into your flask of all of the Green River samples. And so using this, we know how many moles that were present. We know what the volume of that sample was. Moles divided by liters gets us to our molarity. And so however many moles that you calculated in the previous calculation, however many moles of O2 that you had, we're just going to divide that by this volume, by our 30 milliliters, 0.03 liters, and that's going to tell us our molarity of O2. So now we're getting somewhere. This molarity is a concentration, right? And that's ultimately what we're looking for. We want the concentration of O2. So this now gets us to at least a concentration. Tells us what the molarity of O2 was that had to have been present. We typically, however, report all environmental concentrations in units of parts per million. And so we need to convert our molarity over to parts per million. Fairly simple calculation. Parts per million is equivalent to saying milligrams per liter. So all we need to do now is convert moles per liter, so our molarity of O2, so whatever you calculated in the previous calculation, that's your molarity of O2 in moles per liter. We just need to convert that over to milligrams per liter. And so we're first going to use our molar mass of O2, 31.998 grams per mole. That tells us how many grams per liter had to have been present. And then we just convert grams over to milligrams, gets us to milligrams per liter, which is our ppm of O2. And that's our final target. That's ultimately what we're looking for. And so I know this is a lot. This is a fairly complicated process. It's something that we haven't looked at before. Actually, I take that back. We have looked at all of this. All of the calculations that we're doing in this lab are calculations that we've done in lecture. They're calculations that we've done in lab. The biggest difference is that rather than looking at the stoichiometry of just one reaction, you're looking at the stoichiometry of a series of reactions. But it's the same exact process. You're still using your molar ratios in order to compare. We're still converting grams to moles. We're still doing all the same things that we have been doing all semester long in both lab and lecture. We're just putting a lot of it together now, and we're looking at them in somewhat different ways. And so the idea of this pre-lab talk is to give you some of the background. We talked about the importance of dissolved oxygen in the beginning and why we're looking at it, and then walked you through a lot of the calculations. It would not surprise me if you need to go back through this again. You might be sitting there at this point and still scratching your head going, what? I really don't get any of that. That is okay. We're putting things together here in ways that you have not necessarily seen or thought about before. If you're still a little bit confused about it, that is okay. Read back through your lab manual. Watch this lecture video again. The more you watch it, the more you see what's going on, the more things will make sense. And so, like I said earlier, the idea behind this video is really to give you a lot of the background and to introduce you to how you're going to do your analysis and perform your calculations. Your instructor will be talking about how to actually perform the experiment itself, some of the details involved in how you're going to run the titration. And so in the meantime, I hope this was helpful, and happy titrating, everyone. Have a good lab.